as Ala said, I'm not a member of the core, but a user of the core. So was asked to give a talk to kind of provide that perspective. So I'm going to really be talking about metabolomics data in the context of broader tools that I've used, and I'm not really going to be talking a lot about exploratory aspects of the data or looking at the large data sets, but more leveraging the data from a very hypothesis-generated, focused analysis. I really haven't yet gotten to the exploratory stuff, which I intend to. The data sets are very deep, and I hope to get there. And hopefully this afternoon's talks will inform some of the analyses that I want to continue to do. All right, so I'm actually looking in bipolar disorder just to introduce the disease for those of you might not be familiar with it. It's an illness that it has a prevalence of about 2 to 3% in the United States in adults. So you probably know somebody with bipolar disorder, whether or not you do. It's a pretty devastating illness that cycles between manic and depressive episodes. And I'm not talking about mood swings. Everybody has mood swings. We're all on that continuum. But real depressive episodes where they can't get out of bed for a week or two, and manic episodes where they might not sleep for a week, and they might drain their bank account and go out and buy a Corvette, serious financial and social consequences. So, like I said, it's a pretty devastating illness from that perspective. But to put it in the context of the metabolomic and metabolic interest, about approximately 50% of individuals with bipolar also get metabolic syndrome, very high rate of diabetes, cardiovascular disease. So there's this interface between mood and metabolic disorders that goes both directions. That's really been my interest over the last several years. So for those of you who remember the 2 eggs in the frying pan, this is your brain commercial. So this is what we think of as our brain, and neuroscientists, which I used to be, but I kind of consider myself a recovering molecular biologist. Neuroscientists used to, or really focus on this aspect of the brain, this neurochemical signaling aspect. This is the business end of the brain. Neurons talk to each other through these vast networks across these small synaptic gaps. This is where the chemical signaling happens that are transduced into electrical signals that carry it further. But the brain also has this underlying infrastructure of vascular system. And when you think of the brain in this context, it's easier to think of it, as an interface between metabolic health, how the metabolic health of the body can influence the metabolic and functioning of the brain, because it's really an energy and metabolic hog. The brain uses far more than its share, tenfold its share, excuse me, by weight of your blood supply, the cardiac output, the oxygen you breathe, the blood sugar. A lot of it goes to support brain function. If you don't have a healthy vascular system, you probably don't have a healthy neurovascular system, and the whole thing is compromised. So in this context, I think it's easier to think of mental health disorders and metabolic disorders as related. So also the brain is in two-way communication with energy storage systems, with immune system, with cardiovascular system, and the emphasis on the two-way. Again, I think a lot of neuroscientists think of the brain as this master organ atop that gives commands to the body, but the body's feeding back to influence the health and behavior of the brain as well. And again, that brings it into this metabolic context. So in this context, I really became interested in the role of diet in mental health about 5 or 6 years ago. And this is just a review paper. There are several now that puts it into context that diet matters when it comes to mental health. So this was a study of several thousand people in England where they prospectively evaluated and tracked diets and then looked at the risk of developing depression 5 years later. Turned out that a whole food dietary pattern was protective, and a processed food dietary pattern put you at increased risk for mental health after correcting for all of these things in the lower left, including employment grade, education level, socioeconomic status, which obviously correlates with dietary intake as well, correlating with pre-existing diabetes, heart disease, exercise, all of these things. So even after adjusting for all of these factors, diet seems to play a role in your risk of developing a mood disorder, in this case depression. Here's another paper that has kind of a provocative title from a few years ago, Should Depressive Syndrome Be Reclassified as Metabolic Syndrome Type 2? Highlighting the fact that they share a lot of underlying physiological dysregulation. So mood disorders, as well as disorders of altered insulin sensitivity, share perturbations in insulin signaling, inflammatory state, increased reactive oxygen species, the stress system, which is controlled by glucocorticoids in part, is dysregulated. So there is this interface. So the last few slides I've just trying to establish that interface. So my focus has really been on polyunsaturated fatty acid metabolism, and there's a reason for that. There's a growing literature set that's actually not that new anymore, where epidemiological studies have found that associations between omega-3 fatty acid intake, which is omega-3 is a polyunsaturated fatty acid, which I'm sure you've heard of, omega-3 fatty acid intake, which is very high in fish. There's these correlations between societal intake of omega-3s, primarily through fish, and rates of depression, also rates of bipolar illness. Those are just correlative. You can't draw causative relationships from those, but it's an interesting observation. And these are very, very large studies. Also, tissue concentrations of polyunsaturated fatty acids have been found to be lower in people that attempted suicides relative to those that did not. Getting into the more mechanistic stuff, if you deplete an animal's diet of omega-3s, you alter dopamine and serotonin signaling in the brain, which we know underlie some of these mood disturbances. Human postmortem studies have found differences in some of the enzymes that are involved in polyunsaturated fatty acid synthesis in the frontal cortex of both bipolars and schizophrenics. The last point here, that some of the neurochemistry, polyunsaturated fatty acid neurochemistry, whereby they're cleaved out of the membrane and turned into signaling transduction molecules like in the IP3 pathway and things, those are targeted by some of the drugs that actually work in bipolar illness. They might be the targets, but they are targets. I mean, these drugs are very pleiotropic and target a lot of things. So there's a lot of reasons to suspect that polyunsaturated fatty acid may have a role in mental health and mood disorders, and it's kind of been my focus. So just to orient everybody that may not be familiar with this kind of the oversimplified polyunsaturated fatty acid metabolism. So up top there on the left is linoleic acid, LA, and on the right is the alpha-linolenic acid. So the omega-6 pathway on the left, the omega-3 pathway on the right. Those top two are essential fatty acids. You have to get them from your diet. You do not have the machinery to make them, and they are required for life. So you've got to eat them. From those, you can make everything else through these parallel pathways that share enzymes. So they're desaturated and elongated to these longer, more highly saturated forms. You can also get EPA and DHA directly from eating marine sources like fish. Fish oil supplements are pretty much EPA and DHA. So you can get those directly as well. And in fact, some people consider these kind of pseudo-essential because these enzymes in humans are not really that efficient. So we don't really do a great job of taking these and making all of these, although there's inducibility. I'm sure that vegetarians who don't eat any meat probably have higher activities of these than people who do. So there's a lot of complexity here. But the other thing I want to highlight is that these things underlie a lot of different physiology. So linoleic acid itself can be oxidized through lipoxygenases to these various oxalams, oxidized linoleic acid metabolites. These have PPR gamma activity. They're involved in a lot of endothelial function and cardiovascular systems. It could be desaturated, elongated down to dihomogamma linoleic acid, which is a precursor to series 1 prostaglandins and have anti-inflammatory function. It could be further desaturated to arachidonic acid, which is the precursor for many of the inflammatory eicosanoids. So depending on which way you go, you get different results. Arachidonic acid is also very important in the brain. It's esterified to phospholipid head groups and stuck in the membranes and neurons. Neurons have very high membrane to membrane ratios. And arachidonic acid is a precursor to the endocannabinoids, which are known to be involved in mood and energy intake and things. So there's a lot of biology going on from eating this. Same on the omega-3 side. These are processed into resolvins, which really have a role in resolving immune function. They're inflammatory, but they're anti-inflammatory by competition with these because they're kind of low inflammatory, so they have this resolution role. DHA also produces some D-resolvins, which have a role in neuroprotection and things. So there's a lot going on, and there's a reason why these are essential fatty acids. I mean, you have to get them. So I just wanted to kind of lay this out because it's going to be the framework for some of the other things. If you look at how our diets have changed in eating these over the years, this is a graph that spans about 100 years. So this has been a cause for some concern for some people. Linoleic acid in particular has increased from about 2.2, 2.3% of total caloric energy in the Western diet in the early part of the century up to close to 8% of our total caloric energy. So about 8% of the calories you eat come from this one molecule. I mean, it's a big part of the diet, right? And that's because it's linoleic acid is, corn oil and soybean oil are mostly linoleic acid, or very high, 40, 50, 60% linoleic acid. So it's very prevalent in our society, which has a corn and soy-based food economy. Right? Alpha linolenic acid, the omega-3, has gone up somewhat, but not as much. The other ones they've varied, but haven't really changed substantially. So because linoleic acid lies atop of that pathway that drives inflammation, there's been concerns that eating too much linoleic acid may be causing increased chronic systemic inflammation, which underlies a lot of problems, right? But there's two arguments on that, and not everybody believes that. I mean, in general, we believe that increasing polyunsaturated fatty acid intake and reducing saturated fat is a good thing, right? It's associated with reduced heart disease. So these increased rates of linoleic acid are associated with lower incidence of heart disease and several other diseases. But in the cancer field, you can find the opposing literature that linoleic acid is a bad guy and is associated with increase in cancer risk. And the jury's still out. I mean, this is a hot debate in the literature. Big-time epidemiologists are arguing this. So I actually have an R01 in submission to try to look much more deeply at this question. And so hopefully I can do that. So one of the first thing I did with the Metabolomics core, then getting to that, was just to do this. I had 27 bipolars that I had plasma samples, serum samples from. Actually, this is plasma, but it was actually serum in this case. And we did just a free fatty acid analysis, just looking at targeted free fatty acid analysis, looking at 22 naturally occurring free fatty acids, which was all the omega-3s and 6s, and then some other saturated fats and monounsaturated fats and things. And looked at correlations within these blood levels and psychiatric outcomes. And I was indoctrinated going into this with the omega-3s are good, omega-6s are bad kind of philosophy, but my data has not supported that. So this column, global assessment of function, is what it sounds like. Global assessment of function is, keeping a job, social activity, how well are you actually functioning in life? Higher scores are better, right? This is neuroticism, which is much higher in bipolars, personality trait. This is depression, and this is mania. Hamilton depression scale and Young mania rating scale. So across the top, I mean, linoleic acid actually, blood levels of linoleic acid are positively associated with better clinical outcomes, or negatively associated with worse clinical outcomes. So that was not what I expected to see. I was expecting linoleic acid's a bad guy, it's not good, but that's not what this data says. DGLA, which is an extension elongation product of LA, just to remind you, so here's DGLA, right? Extension elongation product, which is a precursor to these anti-inflammatory eicosanoids, has the opposing signature. So it's positively associated with worse clinical outcomes, more negatively associated with better. ALA, which is the omega-3, has a similar pattern to LA. So both the essential omega-6 and the essential omega-3 have similar patterns of correlation with these clinical outcome measures. EPA and DHA, which are the things in fish oil, I saw nothing. I saw no association with those in any of these outcomes. And EPA and fish oil has been tried as a clinical trial for bipolar and depression and has had some success, but the literature is very equivocal on that, on whether it works or not. Again, the jury's still out on that. So this was kind of my first insight into, well, maybe linoleic acid's not a bad thing, right? This is the same blood sample, same patients, looking at the metabolic syndrome marker, standard measures of metabolic syndrome, waist circumference, blood pressure, fasting glucose, HDL, and triglycerides. Same kind of thing. Linoleic acid associated with lower waist circumference, lower fasting glucose, lower triglycerides, DGLA, opposite, ALA, not significant in all cases, but looked more similar to LA. So the essential fatty acids, both the N6 and N3s, associated with better mental health and physical health clinical outcomes. All right, that was only in 27 people. So then I started leveraging this longitudinal study of bipolar disorder that we have here, which is a Prechter bipolar study. I'm not going to get into it too much, but there's over 1,000 subjects enrolled in that. I got a KO1 to actually look closer at these people, and was able to recruit 166 of these folks, controls and bipolars, to subject themselves to a week-long dietary analysis. So they kept a food record for a week, gave us a fasted blood draw, so we could really look more deeply, having dietary information and a larger blood sample. This is just the demographics. Don't really have time to go into it, except to point out that the bipolars have higher BMI, which is expected. That's known. I mean, like I said, they have increased rates of metabolic syndrome and diabetes, so they have more obesity. Some of that is probably secondary to medication use. So looking at the dietary data, this is not focused on polyunsaturated fatty acids. This is focused on everything: amino acids, fats, sugars, carbohydrates, everything. But what popped out was all fats. So the bipolars were eating significantly greater of a couple of saturated fats and significantly less, a lot less, like 40 to 70% less of these long-chain omega-3 and omega-6s. So lower polyunsaturated fatty acid intake, which is probably correlated with socioeconomic status and not eating as much fish and things. So I'm not saying this is driven by the disease, but it's the reality. All right. So mapping this back onto bringing in the metabolomics data and the dietary data and mapping it back onto this pathway that I showed you, these things in yellow, the bipolar subjects were eating significantly less of. These things in blue were lower in the plasma, of fasted plasma of bipolar subjects. Right? So even though they weren't eating less linoleic acid, they had less linoleic acid in their plasma. They had less of these oxidized metabolites, less pro-inflammatory and anti-inflammatory prostaglandins, higher levels of DGLA, which in the previous data was associated with poor outcomes, and they also had lower levels of EPA. So LA, which was associated in the early studies with better outcomes, was lower, and DGLA, which was associated with worse outcomes, was higher. So exactly the opposite of what you would want the profile to look like, right? And then just an interesting association here, splitting them into healthy controls and bipolar individuals, looking at dietary intake versus plasma levels. You expect to see some associations between what they're eating and what shows up in the blood. And that's true for linoleic acid and on both sides for healthy controls and bipolars. Interestingly, more linoleic acid intake associates with less DGLA, even though it's an extension elongation product. So there's some feedback inhibition going on, and and that has shown up in other literature as well. I didn't know that when I first saw it. I thought it was kind of weird, but it has been seen before. But this is interesting. So EPA and DHA, the things that you get from fish oil, controls. If you're a control and you eat more shows up in your blood. But if you're bipolar, there's no correlation between what you eat and what shows up in your blood for these long-chain omega-3s. I mean, like no correlation. So that's, I don't know what's going on there. And still trying to explore that data. All right, so going back to the psychiatric stuff a little bit. So these are the three: linoleic acid, DGLA, and EPA were the three that we found different in blood of bipolars relative to controls. LA and EPA were lower, DGLA was higher in bipolars relative to controls. So using mediation analysis, which I think Shervin I didn't see Shervin's whole talk yesterday, but he might have talked about mediation analysis. We asked whether or not blood levels of these plasma levels explain the effect of being bipolar on these mental health measures. And in the case of LA, the answer was yes. So, bipolars obviously have worse clinical outcome measures, and it was explained by these by lower blood levels of LA, partially explained in this one case by higher levels of DGLA. But when you looked at EPA, even though EPA was lower in bipolars than it was in controls, it didn't correlate with any of these clinical outcomes. So it didn't explain the clinical outcomes. So really the bottom line is everything seems to be pointing to the omega-6 side of the pathway and dysregulation of linoleic acid, not the omega-3 side of the pathway, which has really been the focus of a lot of previous research and promoting use of fish oils, which I'm not promoting the use of fish oils, but I don't know that the data's there yet. All right, this is a busy table just looking at the extension to cytokines. So again, we saw that associations between dietary intake of linoleic acid and cytokines in plasma were different in controls versus bipolars. So it depends on who you are. I mean, the bottom line is linoleic acid good for you or not? I think that the answer is it depends on who you are. And a lot of this, which I don't show here, seems to be moderated by obesity status as well. So obese people may respond differently, actually oppositely, to linoleic acid supplement or higher levels of linoleic acid than lean people. And that's actually the focus of the R01 I'm trying to get funded. Just to finish on this, leveraging the depth of the metabolomic data, because I didn't really go into the depth of metabolomic data at all, just really very focused. If you look at dietary intake of linoleic acid and do an enrichment analysis and ask, are there metabolic pathways that have higher level associations with dietary intake of linoleic acid, then this is what pops out. So bile acid synthesis in an enrichment analysis had a highly significant association with linoleic acid intake, and this was moderated by BMI. So this BMI times dietary linoleic acid term is also significant. So the point is that linoleic acid intake affects a lot of different biochemical, biosynthetic pathways, and that various factors, including BMI, moderates that relationship. So I think that's what's missing from a lot of previous studies, and maybe underlying a lot of the equivocal nature of the literature is things like BMI and even genetic variants, which I didn't get into, moderate these relationships pretty extensively, and we really need to start controlling for these things in order to answer the question. All right. So this just summarizes what I said. Plasma linoleic acid seems to associate with better mental health and physical health outcomes in bipolar subjects. I'm not saying that would be true for the general population. The dietary intake of LA and this inverse association with DGLA is interesting, and how the feedback inhibition might be occurring there. LA metabolites seem to be altered in bipolar disorder and independent of dietary intake. So even though they don't seem to be eating less, we see this dysregulation in the pathways. And I can't attribute these things to EPA and DHA, which has been a lot of the focus in the past. So taken together, the metabolism appears to be dysregulated bipolar, and maybe moderated by both psychiatric medications and BMI. I did try to control for psych medications. I don't really have time to get into that, but that's not an easy thing to do. Right. So acknowledgements, so I have a couple minutes for questions. I am expecting a call from the NIH in 15 minutes, so. Do I think peroxidation after the blood draw? Yeah. Yeah, I mean, I don't know. I can't really speak to that. I think you'd have to talk to a true lipid biochemist. So why would that be happening systematically in bipolars relative? Sure. Yeah. Yeah, I mean, certainly, oxidation, peroxidation is a concern in any of the lipid studies. We try to minimize it, I mean, we draw the samples fresh, flash freeze them right away, and yeah, no, but it's a good point, and I mean, it's always a concern, those kind of post collection modifications, right, in anything, and lipids in particular being pretty sensitive to those. But I was very careful to have the plasma aliquoted and flash frozen right away, so none of these went through any freeze thaw cycles. Everything was flash frozen right after draw, and then thawed right before analysis, or right before the Metabolomics people did their thing. So, but it's a legitimate concern. How long it was just diet tracking, not an intervention. So it was a week. So we tracked everything prospectively for a week so that we could associate intake with what we saw. The R1 I have in submission would be a feeding manipulation, but that's expensive. Okay.